this computer. Okay, so I downloaded that zip folder, I unzipped it, and I'm working through this R script right here. I already have it open. I had this date here just because I was timing the script to make sure it would run in a reasonable amount of time. So I'm highlighting all of our library functions to load the packages that we use in this R code. There's some information it prints out. None of these are errors, so we're okay. We set our working directory. I think this is good. This is not going to be true for your working directory. So if you have not yet, go through, figure out where you're working, build that command, and then you can send it over here. So it's exactly the same for me because I'm working where I'm working. Okay. As we're going through this, it's going to print out the plots in this plots window. But if you want to run through it and see those plots in a PDF, you would uncomment this line for PDF and scroll down and uncomment this dev off. Because if you open your PDF, all the plots will print out on individual pages in that PDF. And then when you get to the very end, you can close that PDF and it will be there to see your plots. But we're doing this interactively, and I want you to be able to see the plots in your plot window and in my plot window. So commenting that back out. All right. If you don't have any of these packages, I provide the handy install lines to do that. Just uncomment it, run the line, and then comment it back. Okay, so I talked about how I downloaded these HTC counts for you from the Xena webpage. This is an old version. So the version you would download today is the star counts. They're formatted in a similar way, so you still have to unlog them -1 and then round to whole counts. And then the filtering will work the same. And then you write the table to your filtered raw counts the same. So if you download a new data set, you uncomment all of this, run these lines so that you have a filtered table. But since I've done this already, we're not going to do that. Okay, we read in our filtered counts table. It's a table I provide for you. Apparently it takes a little bit to load. There it is. It has 28,000 observations, 550 variables. It's in there. Not letting me move this. Okay, I will just leave it there. We're going to import the gene annotation information. This is going to convert those ensemble IDs to gene symbols. We're also going to import our phenotype table, which is clinical measures about these samples. This data set, I think it's a lung cancer. It's a small cell lung cancer. Okay. So first off, I think we've done this already. So I'm going to go through the beginning parts kind of quickly. We check out our variable we want to compare. We see there are 3 groups. We only want the 2 groups in the table that we want to compare. So we're going to filter on primary tumor and solid tissue normal. So we want only those 2 types of tissues, only 2 type of samples. Then we filter it a little more to get a smaller data set, just so it'll run faster. You don't have to do this. This is merely to run the data faster. I filter on stage. That brings us down to 65 tumors and 23 normal. I get some variables set up for later. I want to know how many phenotype columns do we have and what is one more than that? Because this is the cutoff where I'm going to split our table later. And then the last processing thing I'm going to do, if we look at our phenotype table and our counts table, the phenotype sample names have dashes, where our counts table has dots. It's because R converts dashes in a column name to a dot. So we want to keep them the same. So we've made this new sample name with dots in it, period, dots. And we're using the G sub command for that, which is going to replace any dashes with a dot in this column. So that's setting up our phenotype and counts table first part. Now we're going to transpose our counts table, flip it to the side. Now in this transposed version, the rows are samples. Rows are samples, and we're going to connect this table with our phenotype table because we need to make sure they have exactly the same samples and the samples are in the same order. So if we merge them, this is going to take care of that. And that's what I do with these three lines. I merge, then I take that row names column, I throw it back out as row names, and then I remove this unnecessary column. We now have a table that has all of our count, all of our sample information at the beginning, and then all of our counts at the end. One big counts and samples table. Now that we have that, the samples are in the right order, and it's limited to only the samples we want to include. There's 72 of them. There's 28,000 genes. Now we're going to take this counts and samples table and split it. We're doing this using column numbers. So I'm referring to our table and I'm saying in this square bracket, pull out the columns from this starting point to this ending point. And I know what the starting and ending points should be because of these variables. The start is going to be that very first gene, the column number of that very first gene, and it's 126. The end is going to be the number of columns in our table minus 3. And I only know to do minus 3 because I look at the end of the table, go all the way over, and I see there's these 3. I don't know if you remember back to when we were doing counts that we talked about the reads that are easy to count. Those are accounted for, they're assigned to genes. But there's reads that are not easy to count, and so they have to be thrown out. These are the ones, the no feature, the reads that align outside of genes. There's quite a bit of these for each sample. Some of them, like here's 3 million, 2 million, 6 million, just map outside of genes. There's reads that are ambiguous. So they map where genes overlap with other genes. So we can't say that these reads belong to one gene or another. We don't know. And then there's not unique alignments. These are the reads that are mapped in multiple places, and they are also not used as assigned to genes. So we don't want those as columns in our final counts table. And then for the samples, we take from our counts and samples table columns 1 through the number of phenotype columns. If we were to go back, we need to go, there's at least 100 of them. Yeah, it's right here. 125 is the end of the phenotype information. And so that's the end of our sample information. This seems silly, I know, but it's the best way I know how to ensure that the samples are the way we need them to be. If there's a way that you would prefer to do it, you're welcome to do it that way. Coding is you write code in the way that makes sense to you. If you want to use a bunch of lines of code to do what you need to do, that's fine, as long as you understand what you're doing and you can come back to it next week, next year, and understand what you did. Okay, our all samples table. Did we run all counts table? No, we didn't. Okay, so now we have a counts table. It's on its side still, and a sample table. I'm going to clean up our windows here. I have some extra code here because when I do this code on the supercomputer for other workshops and reasons, it takes a really long time to run, even with only 72 samples, when I get to this estimate dispersion step. So when I'm doing it on OSC, on the supercomputer, to show other students, I add in this chunk of code. You don't have to run it. Just watch what I do. And what this does is it's identifying samples in our data set that have tumor and normals present in the data. And there are only 7 of these. See? Yeah, these 7 samples from our filtered sample data table are the ones with both tumor and normal. If you wanted to do a paired tumor normal analysis, this is what you would boil it down to. And there's a stronger, more elegant statistical test you could do. It's a paired statistical test. Boop. I talk about it in the comments down here. If you're interested in a paired tumor normal analysis, check out this link, read about the multi-factor designs. But the basics of it is you're adding in an additional correction in the statistical test that will correct for samples that are coming from the same patient. So this is what you would change if you wanted to do a real paired analysis, but we're not going to do that today. We're just treating these as completely independent samples. Comment. I also don't need this. We're doing that over again. It's fine to do over again. Oh no. No, it's not actually fine to do over again. I'm going to run this again because I filtered my table and I don't want it to be filtered. All right, so now even if we do it over again, it's going to be fine. Let me just check. I might have uncommented. Yeah, I've uncommented more than I should. I'm going to comment these out and then run it again. I'm confusing everybody, I know. Okay, so I'm running this chunk over again. Okay. I keep this commented out because it's just extra in case I'm running on OSC. So we merge our sample and our counts information, make sure we have the same samples in both tables, and then we split them apart. And one important thing I haven't talked about yet is we factor our important phenotype column and we set the levels so that tumor is our primary group and normal is our control group. Okay. So since we have our counts table and our samples table, we're going to take an initial look at the data. And I think I did this already last lecture, but we put our counts in our sample in a DE-seq2 object by first we transpose our counts table with this T function, set that to counts data, our column data. So this is describing our samples. That's the all samples table. Okay, no comments. And then since we're just taking an initial look at the data, we're going to set the design to 1. Normally, the design is what are your groups you're going to compare? What is the column that holds the groups you're going to compare? But in this case, we just set it to 1 because we don't care about groups just yet. Then we apply a normalization to this data set. It's called the variance stabilizing transformation. It's going to normalize and it's going to log transform it. There's an alternative that's like this in DSeq2 called rlog. You can check those out. It does this a very similar thing, but it takes a lot longer. It will transform and log. VST is the faster version, so I use this. Along with this, I'm going to pull out the normalized expression into a table. We're going to use this later, but this is normalized expression. If we wanted to plot something, we would use this. Okay, so the big thing that we want to look at with this bit of code is a PCA plot. So this performs a principal components analysis that takes the 28,000 measures that we have here. Right now, each one of those 28,000 genes is a dimension in our data set. And principal components analysis is going to consider all of those measures and smush it down, condense it down into a few dimensions of variability. This is a dimension reduction analysis because we're smushing all the measurements together into the main sources of variation. And what we plot in a PCA plot is we take that very first line of variance, the first PC, in this case, it is 27% of the variance in the data can be explained by this dimension in our data. And then the second one is PC2, and it can explain an additional 11% of the data. Behind the scenes, there are as many of these PCs as you want to. Each adding another one will explain a smaller percentage of your data. So as you go down, you'll get less and less added on. So most of the information is in PC1 and 2. And the neat thing about these measures is when you plot them, we plot PC1 on the x-axis and PC2 on the y-axis for every single sample, we can get an idea of how similar or different our samples are to each other. We have them shaded here based on our group information. So you can shade them based on whatever column in your phenotype file you want. Like here, I've done new tumor event after initial treatment. We shade them based on that. The PC's values are exactly the same. We're just changing the color. But what we're interested in is tumor versus normal. And there's two things I note from this plot. The first is the normals cluster away from the tumors. There is separation. Even though it's not very much, there's some separation here. There are clear clusters that differentiate the two groups. It's the first thing. The second thing is the normal samples cluster sort of tightly together, while the tumor samples, there's a lot more variability across the tumor samples. If you know that catchphrase that the James Cancer Center uses, it's like no two cancers are alike, or there's no routine cancer. We see that with this PC plot because there's a lot of differences across all of these tumors. Even though they are more like tumors than they are like normals, there's a lot of variation here within the tumors. Okay. So that's a PC plot. It's the first thing I look at if I'm doing bulk RNA-seq differential expression analysis. This gives us an idea of how well this analysis is going to go. You have two distinct clusters here. You're going to get a lot of differences when you look at your differential expression results. If you have something that looks like this, where there's not clear clustering of groups, if we compared yeses versus nos here, I would guess there would not be genes showing as differentially expressed between these two groups because they're too similar. They completely overlap in every way. Now, if you're doing some sort of differential expression analysis, like you're comparing patients who had a new tumor event versus patients who did not have a new tumor event, in this case, I would consider removing these normal samples because it makes more sense to compare tumor versus tumor in this case. We're comparing, were there new tumors after treatment? So I would compare tumors that did not recur versus tumors that did recur. So as you're thinking about building your own differential expression analysis, think about what group you're comparing and how you need to filter your data set to best represent those two groups. Because you want to sort of minimize the heterogeneity in your data set so that those sorts of measurements don't cloud out your results. Because these normal samples, they could cloud out the differences between our yes versus no in tumor samples. So be aware of that. Okay, so we've prepared our data. We've taken an initial look at our samples. Something to think about at this PCA plot is, are there any samples that are outliers? I know in a different setup, I've done this. One of the tumors falls over here in the normal samples. And that's a big red flag to me because that tumor, it's either misclassified or it has a big chunk of normal tissue that was sequenced along with it, or there's some sort of error here if one of our tumors falls within the normal side. So I would remove that tumor from the analysis because it's not clustering correctly. Okay, so now we've done all of that preparation. We're getting to actual differential expression analysis. This is the easy part because it's like we could boil it down to two commands, but I break it out just because it's makes more sense to see all of the different commands. First, we put the data into the DE-seq2 object. I get an error. I get a warning. The warning is fine. So it's saying levels of factors in the design contain characters other than numbers, letters, underscores, and dots. This is because we have spaces in our group names. And we're just getting a warning about that. This is a warning. It's just a message. It's not even a warning. It's just letting you know, are you sure you want to do that? It's okay if you do it, but it's it's letting you know. But now we have a DSeq2 object. We're telling it, we're grouping it on sample type. This is our tumor normal. Here's our counts table that is transposed. So the rows are genes the columns are samples. Our sample table has rows as samples. And DSeq will check to make sure the row names of your sample table equal the column names of your counts table. And it will I think it will just fail if they're not equal. Okay, now we're doing these three commands, which can be replaced with that dseek2 command. This command replaces all three, but I like to break them out. So first, it's going to normalize the sample to sample differences. We talked about that last time. It's going to estimate the dispersion. So it's going to look at the variability in the gene expression and squish it down to the mean. And then it's going to do that negative binomial walled test to calculate our p-values. Along with this, we talked about it last lecture, it's going to adjust for the multiple testing using the Benjamini-Hochberg correction. If we click here, it's going to talk about that. Oh, okay, we are done. I don't know. There's so much information. You can read about the statistics behind it. You can read about the fold change. You can change the different options. I would not change anything unless you know what you're doing. Okay. And then this very last command is going to finalize the results. Save it in a res object. What is it? This takes a bit of time too. And this is taking more than the dispersions took. If you wanted to avoid this message, back up here, we could have renamed our groups to just normal and tumor, but I don't mind the messages. Okay, now we have a large DE-seq results object. It has a bunch of stuff packed into it. It's a fancy way of storing data. We can get a summary. We tell it what is our adjusted p-value cutoff, and it tells us out of 28,000 genes, we cut it off with an adjusted p-value less than 0.05. 7,700 of them have a positive fold change at that p-value cutoff, and 5,000 of them have a negative fold change at that cutoff. We know since we set the levels up here, we know that a positive fold change means it's higher in the tumor, and a negative fold change means it's higher in the normal. And I'll demonstrate that later because I don't trust my level assignments. We can pull out the results table from this res object because that's annoying to have it in an object. Now it's a data frame. It has the average of this gene across all the samples. It has our fold change. It has some statistics columns, which we don't care about. And it has our p-value and our adjusted p-value. It has ensemble gene names, which are annoying to think about. So we merge this with the gene annotation table that we imported at the beginning. We set the row names as row names, and we remove that column. So this is a more informative results table. It has our same differential expression results, and it also has our gene name. Now, there's something unique about DE-seq2 that, here's one. If, I talked about this last lecture, if the counts are so unstable that it thinks that this statistical test is inflated and it's not a good test, it's going to set the p-value to NA. So these have been filtered after the fact based on DE-Seek2's consideration. So we're going to remove those from our table. We're going to subset our table where this p-value is not NA. Let's see how it changes in number. We're at 28, 135, and now it goes down to 26, 251. This point, we're going to save our table just so we can come back to it if we need to. I do a write table of this data frame. This is our text file we're saving it to. We're going to include row names. We want call names, so we're going to set that option to NA. I do not want quotes, and I'm going to separate it with tab. So now in our folder, we have a text file right here. Looks just like it did in R. So that was differential expression analysis from here to here. Now we're going to visualize this a couple of different ways. The first way is a volcano plot. It's using an R package called Enhanced Volcano, which makes very nice plots just automatically. We tell it the column name from our table that we want to use for the fold change. On the x-axis is fold change, and the y-axis is the negative log of the adjusted p-value. We're telling it use the adjusted p-value here for the y. And doing this negative log 10 makes the smallest of small p-values the biggest. So what is most interesting to us in this volcano plot are these two peaks of the volcano. It's called a volcano plot because it looks like a volcano. And these are the parts. They have the smallest p-value and the largest fold change. Here I am including labels of the gene symbol. It's going to label as many genes that will fit comfortably. There's still some dots here that don't have gene names. If you have genes that you really want to highlight, there are ways of only labeling those genes. If you want to include as many genes as you can with lines to the dots, there's ways of doing that too. The Enhance Volcano GitHub. The website for Enhanced Volcano is very useful because they go through nearly every option that you could want to plot with this. Different shading. See here they label selected genes. So their website here, their user guide is very nice. Everything you might need to know about plotting a volcano plot. I mean, if you really wanted to, you could plot a volcano plot with ggplots. You could tell it, use the log2 fold change on the x-axis, do some math on your own to transform this adjusted p-value, draw a scatter plot with ggplots, control the shading however you want with custom cutoffs. Here we can specify our cutoffs for p-value and fold change. Like if I wanted a fold change of 2 and a p-value that's very small. Where's my plot? Okay, there it is. So these cutoffs for lines, they've moved a little bit just because I changed the cutoff. So this plot, it helps us get an overall view of all the genes that we measured in the data set. It can give you an idea of, are there more genes upregulated than downregulated? Is there sort of an imbalance in the directionality of our gene expression? How might we want to set our cutoffs? Well, maybe we want to increase the fold change cutoff more than we increase the p-value cutoff. You can think about those things with your volcano plot. Now we're going to talk about heat maps. Let me check for comments. Yeah, no comments. The next plot we're going to talk about is a heat map. Let's check out how many genes we might have as significant at different levels first, because we need to select the genes that we're most interested in visualizing. First round, I did a fold change, either up or down, using this absolute value of 1. So either greater than 1 or less than -1. And I require that the adjusted p-value is less than 0.05. I know this is going to give us a lot of genes. Yeah, nearly 10,000 genes. And if we plot that on a heat map, the heat map's going to complain. So we're going to filter a little more strictly. I'm going to set the fold change cutoff at 4, so way out here, and the p-value adjusted at whatever this is. It's a small, small number. That gives us 700 genes. So that's more reasonable. We can work with 700. I'm also going to get an account of how many samples we have, because I'm going to use that. Okay. So first thing, I'm going to take our significant results table that we just made up here. This is just our differential expression results with their genes. And I need to pull that expression, the normalized expression that we put into MAT earlier. We're going to merge those based on ensemble gene name. Significant expression. So here we have our annoying row names. We have our expression of all of our samples. I need to tab over samples, more samples, more samples. Okay, and then we have differential expression results here at the end, which we don't want. We want to just get expression values. And we want to set the gene names to be our row names. So that's it's another annoying part. If we try, let's see. If I try to do this right now, I'm going to set the gene column to be our row names. Oh, it works. Well, this doesn't demonstrate my issue at all. Okay, so there are some gene names that exist multiple times in our data set. This gene name, Metazoa SRP, it's some sort of non-coding, I don't know if it's a bacteria or what, but it's something present 6 times in our data set. This is true with U8 and yRNA and even whatever this one is. So I've gone through this a couple of this code. I've used it over and over again, and I always run into the problem where I get some of these genes in the results table, and when I try to set the gene column to be our row names, I get an error and I have to clean up the table. But apparently with this specific filter, I don't get that problem, but I'm still going to remove them. It works even without that. So I'm unproving my own point. Okay, we're going to clean up our table. We're going to select the columns that have samples. So that's why we had this in sample variable saved, because we know we have 72 samples. We want these first 72 columns. There we go. And so this is our finalized expression table. Now to plot in a heat map, let me show you how badly it would look if we did this. If we just plotted the normalized expression, it would look a little funky. It's plotting normalized expression. Expression goes from 0 to 15 here. And since each one of these genes, they're sort of on a different scale. When we plot this, it doesn't look very nice because it's not meaningful to us. If we look like at this PDK gene, it's sort of averaging probably around 8 or 9, where a different one, let's see, this one, Hawks A11, it's sort of averaging a little bit lower, around 3. So each gene has its own range, and we need to get these genes sort of standardized. And what we're going to do is we're going to center the expression of a gene onto 0. So average expression for PDK4 is going to be 0, and then it's going to be scaled either way. Average expression for Hawks A11 centered onto 0. And we do that here with this scale function. With first, we take our significant expression, we flip it on the side because scaling only works on, I guess it's on columns. So we transpose it, we scale it, then we transpose it again to get the scaled expression that we want to use for our heat map. The nice thing about this heat map application that we're using is we can build up annotations. So this is sample annotation, and it's going to shade any sample that's a normal with gray, any sample that's a tumor with dark green. And this will make it a lot easier for us to say, are the samples clustered only within their groups? So we save that heat map annotation and we plot our heat map. Okay, I don't know about you, but this heat map looks a little nicer to me. It's still not that impressive, but it does look nicer. So average expression for a gene is going to be 0. Above average expression for a gene is going to be shaded in red. So these are above average in our normal samples. Below average expression will be shaded in blue. The samples are columns here, and the samples are clustered based on the expression profiles of this select set of genes. The genes are also clustered over here on the side, and that's why we have on the top, there's a cluster of genes that are expressed more highly in our tumor samples. You can see there's more red all across these. Where the bottom chunk of genes, they're expressed more highly in our normal samples. We can look at the sample clustering. We see the color of our samples. They're divided correctly. We have our cluster of normal samples right here. We have a few clusters of tumor samples, and I want to bring your attention to this cluster of tumor samples that are right next to our normal samples. Do you notice how the expression of this upper panel of genes, it's overall more blue than red, where all of our other tumors, they're more red, where for this one cluster of tumors, their expression for these genes, I would propose, looks a little bit more like our normal samples because they're more blue up here at the top and they're more red down here at the bottom. Just like these tumors that are strikingly blue at the top and strikingly red at the bottom. Going back to our PCA plot, I would guess that those tumors that clustered in their own cluster, apart from the other tumors, are these that exist closer to our normal samples. We could check that out, but that's what I would suspect. There is a population of tumors here that are slightly more normal than tumor. Now, is this because the tumors are less aggressive or because they have a higher proportion of normal sample that might be attached to them? We don't know. Or these could be the tumor samples that have paired normals in the data set. We're not accounting for that in our analysis. So, oh, there's a comment. Yeah, if we subset the genes more strictly, this plot might be clearer, like even more striking. I know I did this morning with the high school students, and I limited it to the 7 samples that have both tumor and normal. And that heat map was beautiful. The genes were really strongly red or blue, and it gave us really clear symbols on the heat map. Now, it wouldn't make any difference plotting the expression. This is always going to look sort of poorly just because they're all on the same. I cannot move that. Okay. They're not on the same range. So this is never going to be that good. But yeah, this I consider this a success because the sample the I can't get to it. The the tumors all cluster together and the normals all cluster together. It's just there is some difference in the tumor. If we look at these nodes, yeah, the tumors are all on the same node. So even though they're slightly different, they're still tumors together. And there's different ways of clustering too. I would assume complex heat map you can try the different ways of clustering. We could spend a whole class on clustering methods, but yeah, this is just the default. Complex heat map is another one of those GitHub. Yeah, it's on there, but they have a really great book on how to use complex heat map. And it goes through all of these examples and different types of heat maps and annotations you can add. There's things I don't even know about. You can have interactive complex heat maps. I've never done that. Doesn't have much information. Okay. But yeah, this goes through all of the things you could do with complex heat map. And you could read this and it would take you months to read through it. But what I do is if I need to do something, I go look at the example of what I specifically want to do, like adding in annotations. You can have, well, so here we have column annotations. You can also have row annotations. So maybe you're plotting some particular keyword term and you want to mark that some of these genes are from pathway A and some of them are from pathway B. You could have row annotations here. It's just whatever you want to build up to prove your point in your plot. You can also you can plot whatever gene set you want. If I wanted to plot this, let's see if this works fairly quickly. Okay, our SCA. Oh, I can't do this right now because of duplicate row names. Well, it's one I didn't get. Yeah, there's a bunch of them here, so I'm not going to do that. If we include more genes, we run the risk of having more duplicate row names. So I can't demonstrate that. Now, I have a column title here that controls the name of this plot. It's actually not valid because I did change these values. This should be 4. So I am describing what cutoffs I use here. If I change those cutoffs, it's good form to change that in the title so nobody gets confused. There we go. Now it's a 4. So make sure your title is appropriate to what you're actually plotting. Okay, so the last plot I'm going to talk about here are box plots just to check a few genes. So I am merging my significant expression with our sample information. I'll show what this looks like in a bit. Significant expression sample. Which one is this? This one. Okay, so I'm attaching all this sample information to our table of significant expression. So these genes back here, they're the genes that were picked out because of their fold change and their FDR, their adjusted p-value. And I added in sample information because I want to plot box plots. I ask, what is the gene with the largest fold change? So I could do this manually. Actually, I don't know if I could. I could go to my results table. I could sort this. Okay, yeah, Hawks D13 and our significant results table has the biggest fold change. The one with the smallest fold change is this RP11 whatever. We can go the other way. RP11 whatever. So those are the ones I'm plotting in my base R box plot. This is the positive gene, and it's higher overall in our tumor samples. That's what we expected it to be. Now, our most negative gene is higher in our normal samples. I do this because I do not trust my ability to set a factor, because I always get mixed up which should go first and which should go second. If this doesn't match up with what you hoped it would be, then you would go back up here where we factored our sample, factor, and switch these levels around. Apparently, we put our control sample first and then our case sample second. It seems backwards to what I would have picked. And that's why I always get it wrong. So this is just a quick check, is what we're expecting, plotting out how we wanted it to. Maybe you were writing your manuscript and you want to highlight the expression of your favorite gene. And so you could do that with a quick box plot. You could show my favorite gene expresses more highly in the tumors than in the normal. Okay, so that is our script. Before we get into functional enrichment analysis, I want to do something that I did this morning with my high school students. So we did differential expression in the correct way with DESeq2, which calculated our fold change, and it normalized our expression, and it calculated a p-value. But what does that mean in real life? So I'm going to do differential expression by hand in Excel. It's a silly thing to do. But first, we need to know how many reads exist in each sample. We do that by summing the column. So sample T1 has 31 million reads. Sample N2 has 93 million reads. I'm going to divide each one of these by a million just to make it easier. Okay, So since we know how many million reads we have per sample, we're going to calculate counts per million. So this is our normalized column. We have our count. We divide it by this sum, read sum for our sample. We do the Excel shortcut. We want it to be that. Oh, I am in the wrong space for this. Here we go. This divided by this. Function. Okay, we get this. So here we take 1427, divide it by 31 million. And this is our normalized expression value over here for that gene in that sample. We paste that across. So let's double check our Excel math. We are taking this count and dividing it by the million reads for this sample. So that works. We fill in our table. We average these three, average these three. Okay, so now we have our tumor average and our normal average for all of our genes. We're going to calculate a fold change. Maybe we should do a log fold change. Log 2 fold change. And we want to set this so that if our tumor average is bigger than our normal, it's going to be positive. If our normal average is bigger than our tumor, it's going to be negative. So we need to do an if. If this is bigger than this, then log this divided by this to the base 2. Otherwise, negative log of this divided by this. So we're calculating a fold change, and then we're logging it. This is our log 2 fold change. So this is the fold change of the normalized expression levels. And then we're going to do something that we should not do. We're going to calculate a t-test between our two groups. Now, we've already talked about the correct statistical test for differential expression analysis is the negative binomial distribution. And then we use a Wald test to get our p-value. But we can't do that in Excel, so we're just going to do a t-test to get us a p-value. Okay, so we get our 3 tumors, we get our 3 normals, we tell it 2 tails, and we want equal variance. So this is our p-value of what is the significance associated with this difference. We could, let's see, let's do a negative log 10 of this p. This is just a column name. Okay, equals of this base 10. We have to negativize it. It's a completely made up word. Okay. To make this easier, I'm going to put it back here. Okay, so we have our log 2 fold change. We have our negative log 10 of our p-value. We're going to insert a scatter plot. Okay, it's way up here. And here is our hacky volcano plot doing differential expression in Excel, which is not recommended, but it gets us something similar to what we just did. And we can kind of think it out. What is DESeq2 doing behind the scenes in a simplified kind of way? Don't save. So that is how we do differential expression with dseq2 in R. The new thing we're talking about today, we have 20 minutes to talk about it, but let's see. I have a new script for that. Okay, we're going to close this R. We're going to talk about differential functional enrichment analysis of our differential expression results. If you go to where we're supposed to be today, but we're horribly behind. Let's see. Okay, that is, yes, last week. This is today. Functional enrichment, download this hands-on. We're going to go through it either today or on Thursday, but okay. Hide floating meeting controls. Okay, so this is the last topic that we have to talk about in our bioinformatics section. We've gone through bioinformatics, genomic databases, sequencing. We saw how to download publicly available data, run differential expression analysis, and now we're trying to find out whether there are biologically meaningful trends in our list of genes that maybe we can connect back to our original hypothesis. So we're going to talk about some methods for functional enrichment analysis, how to format the input data to run analysis, and to interpret results. So first we have to talk about gene terms, functional gene groups that people who develop databases for these have decided to group genes into terms. One of these databases is called Gene Ontology or Go Terms, and they group genes based on function, relationships, classes, and concepts into these three main groups: molecular function or molecular activities, cellular component or where the genes are active, biological process, which is more biologically minded and what is happening in a wider sense. Now, people talk a lot about pathway analysis, and I would say gene ontology terms are not pathways. If you want a pathway, it's a defined network, really, more than just a gene term. It's a network, and you're going to find these networks in KEGG, which is the Kyoto Encyclopedia of Genes and Genomes. These are defined pathway maps representing interactions and networks. There's also wiki pathways, which I think overlaps a lot with KEGG. There's disease ontology, but all of these different types of gene terms, let me see if I can link to it. Yeah, are defined on this MSigDB database. I use this all the time to, hey, let's see, search. You have to have an account, which is free. Here, I've done some already. Interferon. Where are all of the interferon related genes, pathways? I can limit it to gene ontology. So here are all of the Go terms relating to this keyword interferon. There's a bunch of them. We can export to, oh, I gotta select some. We can export to a file. I suggest GMT. It's a specific format for gene sets. And it's down here. It's going to have one gene set per row, the name of that gene set, the website, and then all of the genes. And there's a package, I don't remember it right now, but there's a package that can import this into R. I can look that up for anyone who's interested. So I've done things where I create a custom gene set list, and then I do heat maps or I do functional enrichment for just that custom list. What we're going to do together is testing all of the gene terms for our differential expression results. Something that's important to know about gene ontology terms is they are connected into a hierarchy so that there are broad terms. So the broadest is for this one is biological process. It's made up of subterms that cellular component biogenesis is present completely in biological process. So there is a specific end down here at the bottom, and it gets more general. And there's you'll see like positive regulation of coagulation and negative regulation of coagulation. And then you'll just see coagulation, which is made up of the positive and the negative regulation combined. So there is redundancy, there is hierarchy, so that some gene terms are completely present in a broader gene term. This is only true for gene ontology. So you might hear the phrase pathway analysis. I'll be asked, can you do pathway analysis for me? And I'm like, well, what do you mean by that? It's a general term covering anything pathway related. They'll refer to a Go term enrichment as pathway analysis. And I don't like that phrasing. I prefer functional enrichment analysis, specifically overrepresentation analysis, which is where we filter the gene list and we look for an overabundance of our gene term. And I'll show how that works the next slide. Another option is called gene set enrichment analysis. Is there an enrichment of this gene set with a preference to one side, one group or the other, our sample groups? When you do gene set enrichment analysis, never filter the gene list. You only sort the genes. So functional enrichment, filtered gene list. Gene set enrichment, full gene list. People get that messed up all the time. They publish and they still get it messed up. Okay, so first we're going to talk about functional enrichment. This is an overrepresentation analysis. We're asking, is our biological category more represented in our differentially expressed gene list than we might expect by chance? And we do this simply by counting. We take our list of differentially expressed genes. Here we have 60 of them. We see there are 28 of these genes in the gene term. They're in the category. Where there's another 29 of our differentially expressed genes that are not in the category. So it seems like a 50-50 split. So I would predict our category is not enriched. And we also have to consider all the genes we've tested, genes that are not in differential expression. So we count those up. How many not DE genes are in our category versus how many not DE genes are not in our category? And we test this. You can test it with a chi-squared or a Fisher exact test, but this is our measure of statistical confidence of whether this gene term is enriched in our list more than we might expect by chance. And we'll learn about Fisher exact tests and chi-squareds possibly next week when we talk about statistics. But we're looking for whether a gene term is more present in our differentially expressed gene list than we might expect. We also need to do multiple testing correction because we're testing for maybe 1,000 gene sets. There are some what I call copy and paste applications. There's David, GeneCodis, and TopGene is my favorite, I think, out of these three. It's fairly straightforward. With TopGene, you paste in your list of differentially expressed genes. We could pull out our list of 700 or so. If you're doing some sort of targeted expression analysis, you might need to specify your background. The background is the set of genes, this not DE gene list, or it's the entire universe of genes that you are testing. Do you need to provide that background set? If these results are from this bulk RNA-seq that we're doing, it's assumed that all the genes are measured. So normally you don't have to specify a background. Like in our case, we're not going to. But maybe you did a nanostring project and you measured the expression of 800 known cancer-causing genes. And that's the only thing you measured. You found 25 of them were differentially expressed. So your input gene list is those 25 genes, and your background would be the 800 genes that you tested. So think about when you're doing this, whether you need to specify a background or not. You can pick in top gene what gene set terms you might want to use. There's go terms, there's pathways, there's drug diseases, there's genes targeted by microRNAs, all of these. You can specify what sort of correction you want to do and what p-value cutoff you want to do. I usually never change any of these options. If you're going to change them, you should have a good reason for doing it. And then these are your results. These list the terms that were enriched to your data set, the corrected FDR p-values, and here you can even click and see what are the 7 genes from your input list that connect to this gene term that is enriched to your data set. Here's a plot. Now this plot is very confusing. I, hopefully I can understand what it's doing again, but each line has its own um, axis. So let's see. The red one, genes in annotation, goes with this numbering up here. So there are about 450 genes in the annotation. The genes in common in blue goes with the genes common with training set count. So there's 6 genes from our data set in this gene term. And then there are some p-value lines here. You can barely see them. And they go to this axis down here, p-value, which they're all very small. I really wish this was like a negative log 10 p-value, because then you could kind of see it better. But there's this plot. It's a rubbish plot. So you can look at it if you want. You get to it by clicking display chart. You see this chart. You can also, so there's more enrichments. If you click on show more annotations, it's going to list a bunch of things. This is the disease one, and it sometimes it might give you the disease that you're actually testing, but it's going to give you a lot of extra stuff too that may or may not make sense. This is all based on, is this showing up more than you would expect by chance, but sometimes you're still going to see things by chance. Okay, what I like to use for over-enrichment analysis is this cluster profiler package. You can perform over-enrichment of gene ontology and KEGG. You can also do gene set enrichment analysis, which we're going to talk about in a few slides. But I will show this in the code. But if you're going to enrich gene ontology, there's an enrich go function. You tell it your gene list, you tell it your organism database, what type of ontology you want to run it on. Is it molecular function? Is it biological process? Is it cellular component? You can do all. That can be an option, all here. You tell it your p-value adjustment method. There's some options here. I always use Benjamin E. Hochberg. You tell it some cutoffs, and you tell it what symbol, what type of database your gene symbol is in. So these are gene symbols. You might have ensemble names. You might have entree names. You might need to convert from one type to another. Now, I've explained to you how these Go terms, there's redundancy, there's overlapping. So there's another function that you can use to simplify your results. So you might see where two different gene ontology terms are enriched in your data set, but when you look closer, they have exactly the same genes. So the simplify function will clean that out a little bit. So when we look at our results, this is what it's going to look like. It's going to list some gene ontology terms. It's going to tell us what is the ratio of differentially expressed genes in the term versus all of your differentially expressed genes. It's also going to give you those numbers for the background, not DE genes in the term versus all genes. Here's our adjusted p-value. And then these are all of the genes in our differentially expressed gene list that are connected to the term. There will be 24 genes listed here. If you're going to do KEGG functional enrichment, there's an extra step. I haven't given you the code for it, but if you're interested, I can share that. But we have to convert our ensemble or gene symbols to entree ID, which I use with a function called bitter. I'd have to look up the commands that I use to do that. Here are some plots that we can make with our functional enrichment results. The bar plot, the length will be based on this number of DEGs. The color is based on the adjusted p-value. Alternatively, this is a dot plot where the size of the dot is based on the proportion of genes, and the color is based on p-value. This version that I did right here, this is from a compare cluster where, let me see, hide floating. I have the commands for this, but I split the genes into those with a positive fold change, upregulated, and those with a negative fold change, so downregulated. And I had it do separate enrichments. And this shows us the genes that are upregulated are tagging different functions than genes that are downregulated. So you could imagine our upregulated genes could be oncogenes, could be driving tumor growth and function, where our downregulated genes, they could be, I don't know, but these are higher in normals. Here is a gene concept network plot. It's going to plot the linkages of genes with biological concepts as a network. So each one of these gray dots is a gene. The tan dots are a gene term. And we can see cell-cell adhesion mediator activity connects to structural constituent of cytoskeleton based on one gene. Then there's more connected ones over here. So this is like modules of gene terms. Like here would be one module, here's another module, and then there's histone kinase all by itself. Okay. I will get started on gene set enrichment analysis, but I probably won't get very far. Gene set enrichment analysis uses all the genes, and it's going to rank your genes based on their fold change. It's going to calculate an enrichment score, which makes more sense when I show the plot, and estimates the significance of your enrichment. Let's see. The easiest way to do gene set enrichment analysis is to use, Wait, where's the, I don't have that. Okay, let me show you. Chat. Uh, Confused with the term not DEG with respect to all the genes that are go term but not to all the genes. Okay, so first we have our definition of differential versus not differential. Let's go back up to our square. We can move this. Okay, so and if we just do this versus this, we have some genes that are differentially expressed, so that makes sense. But then we have all the rest of them. Let's say we test 2800 in our example, and we are defining those differential genes as those 700. So we have 27,300 genes that are not differentially expressed in our data set. And those could be connected to a gene term like autophagy. Some of these might be in the gene term and some of them might not. Let me go to that results table. That might illustrate it better. So here we're testing 18,000 genes. 427 of them were differentially expressed. Out of out of all of our 18,000 genes, let's see, 269 plus 24, nope, 269 plus 24. So there are 293 genes in the term microtubule binding. Like if we were to look up in the database, it would probably have around 300 genes. And 24 out of those 300 genes were in our differentially expressed gene list, where, let's see, 269, so that's 8%. 269 out of 300 were present in the total term. I don't know if I'm helping describe this any better or not, but we're dividing all of our genes into 4 groups. And in the first dimension of split, we split them into differential or not. And in the second dimension of split, we split them into in the gene term or not. So that our 18,000 genes that we're measuring, they are populating those 4 categories in our contingency table. I'll try to describe this again next week, and maybe when I describe it again, it will make more sense. But I'm not going to go over any more of these slides today. We will finish up on Thursday. I will stop recording.